All right, so we left off on Monday talking about how light provides energy. And we talked about that light travels as a wave but behaves like a particle, and that light, when a particle of light called a photon collides with something, it's going to impart its energy. We also talked about the relationship between wavelength and energy, that the longer wavelength is, the less energy in that photon of light. So then the next question is, well, how does the plant harvest this energy? So we have an idea of what light is. Light is radiant energy. That's what light is. It is energy. We're like, okay, but how does the plant harness this energy? Because really, we need to convert radiant energy into chemical energy. Okay, the first way to answer this question is basically to talk about the pigments. That plants have pigments that absorb light. Now, some of these are green, the chlorophylls. Some of these are like a yellowish orange, the carotenoids, the accessory pigments. But plants have these pigments that absorb light. And they absorb light differently at various wavelengths. We mentioned this a little bit on Monday, and I said the reason why plants are typically green is because plants have a very specific pigment type that absorb all parts of the visible spectrum except green, right? That light does not get absorbed and it gets reflected back. So these pigments absorb light differently at various wavelengths, and here's what's actually happening. So within the pigment molecule, there are electrons, just like there are in any molecule, right? A molecule of water has electrons in it. A molecule of carbon dioxide has electrons in it. One of these pigment molecules has electrons. And some of these electrons are at a relatively low energy level. And this is compared to electrons in other situations. Okay, so these electrons are stable. They're in a low energy level. They're exactly where they want to be. When you hit these electrons with light, if you hit these electrons that are at a low energy level with a photon of light, those electrons absorb that energy. So again, light is radiant energy. And when that photon of light hits the electron, that energy goes into the electron. And so now that electron starts to move faster. And as that electron moves faster, it jumps up to a higher energy level, goes to an excited state, but the electron doesn't want to stay there, right? If it wanted to stay there, it would have been there in the first place. But electrons want to be in the lowest energy level possible. And why is that again? Somebody remind me. We've talked about this before, and I know you've talked about this in chemistry. Why do electrons want to be in the lowest energy level possible? Yeah, Allison.
Because they're closer to the nucleus.
Yeah, they're closer to the nucleus. Electrons are negatively charged. The nucleus is positively charged. They want access to that positive charge. And so as these electrons absorb the light energy, they jump to a higher energy level, but they don't stay there. And there's basically three options for those electrons. The first option, they drop back down to where they started, right? So they get excited, jump up to a higher energy level. First option, they drop back down to where they started. And if they do, they'll emit light. And typically the same light that they absorbed. So if they absorbed a photon of red light and they drop back down, they will typically release back red light. Okay, so that's one option. The electrons just drop back down to where they started and they release light. And you're like, well, cool, that didn't really accomplish anything. Like, how does the plant use that to create, or not create, but to, how does the plant use that to facilitate the formation of chemical energy? Right? The electron just went back to where it started and re-emitted the light it absorbed. The other two options is where something interesting happens. One is that electron can completely leave its nucleus. So another option is the electron could have absorbed just the right amount of light to get just the right amount of kinetic energy to travel at just the right speed to entirely leave the nucleus of that atom. And where does it go? It goes to an electron acceptor. And so we're like, is this what happens with the electrons that are split or the electrons that are retrieved when we split water? And the answer is yes. The electrons absorb just the right amount of light to get just the right amount of kinetic energy to have just the right velocity to actually leave the nucleus of whatever atom they're interacting with. Okay? And then they can go to the electron acceptor. And now these electrons can start to jump down, lowering energy, fueling the active transport of hydrogen ions, right? Which can fuel chemiosmosis. We've talked about this? Maybe? Yes? Yes. Yes. Okay, so that's another option. The electron can be transferred. It can completely leave. The third option is the electron can drop back down, but instead of emitting its energy as light, it can transfer that energy to an electron in a neighboring molecule. Okay, so instead of when it drops back down releasing light, when it drops back down, it can transfer that energy to a neighboring molecule. But not all of it. Some of it will be lost as light, some of it will be lost as heat, and some of it will excite an electron in a neighboring molecule. Almost like playing a game of, yeah, this analogy isn't great, but it'd be like playing a game of hot potato, where when you tossed your potato, it hit your partner's potato and got their potato excited to toss to the next person. Right? I was going to say it's like playing a game of hot potato, but really that was more like the second one, where the electron moves from one molecule to the next, right? And then that thing's like, oh, I don't know what to do with it, and passes it on until the music stops, right? And hopefully by then the electron's where it's supposed to go. What's the final electron acceptor in the light-dependent reactions of photosynthesis? Oxygen's the final electron acceptor in cellular respiration. The final electron acceptor is NADP+, right? In the light-dependent reactions, it'll take those electrons, take them over to the light-independent reactions, and then the final electron acceptor is what gets reduced during photosynthesis? Carbon. Carbon dioxide. Okay, so here's a graphical representation of this. Electron just chilling in its low energy level, gets hit by a photon of light. That is converted into kinetic energy. The electron gets excited, moves up to a higher energy level, and three options. It can drop back down, emit light, typically the same wavelength of light it absorbed in the first place, maybe a little bit different because some of the energy may be lost as heat and not as light. Or it could completely transfer to another molecule, or it could transfer its energy to a neighboring molecule. Okay? Yeah?
How can an electron transfer its energy and not itself?
So when it drops back down here, it's showing you it's releasing it as light, but it can release it as heat. It can release it as any type of energy that can be absorbed over by the neighboring molecule. It could be light, it could be heat. It could just be, it could just be if that electron actually physically collides with another electron. That's going to be extremely rare because electrons are tiny and this space is huge. But if it physically collided with it, it could transfer energy that way. But most often it's going to be transferring it as light or heat. Yeah, that's a good question. Yeah.
Let's say the electron was on the third layer and it dropped down to the second layer.
Yep.
Released all its energy. What would happen if it went down to the first layer?
So if it went lower than where it started, it can't go lower than where it started. Right? So the electrons are where they are because you have to fill lower energy orbitals before you can fill higher energy orbitals. So the electron can't go down lower than where it started. Yeah. What would happen is if you hit let's say you had the first and the second full, but an electron in the first got hit by the light and jumped up to the third, an electron from the second would drop down to the first to take its place.
And then would that cause the third to jump to the second?
Yes. So it might end up dropping to a place higher than where it started, but it wouldn't drop to a place lower than where it started. Yeah, this is a good question. Unless you had like a perfect storm, like several electrons in the same atom got hit simultaneously, one inner, one outer. That outer one dropped all the way faster than the first one. It'd take like a perfect storm. Yeah. All right. Does this make sense? Our three options? Okay. And so here, just real quick, the structures of these pigments. I just want you to see that these are just molecules, right? Now they're really big, complex molecules, but they're just molecules. And molecules form because these atoms want a fully filled valence shell, right? That's why these molecules form. Why individual atoms form molecules, because they want a filled valence shell. And so most likely these electrons that are going to be hit are going to be those in the valence shell, in the outermost energy level, okay? And so they'll jump up and then typically they'll drop back down, releasing their light to a neighboring molecule, just releasing their light in general. So here's carotenoid, an accessory pigment. It's a little bit different a molecule. The electrons it's exciting are actually in this long chain of carbon double bonds alternating with single bonds. Okay, if you're wondering, like, do we need to be able to do these? The answer is no. This, I just want to show you that they're just molecules. These pigments, they're nothing. I mean, they're cool molecules, but they're nothing like crazy. They're not like some complex molecular machine. All right, so here's just an illustration showing you that plants perform best. They absorb the most amount of light in the blue portion of the spectrum and in the red portion of the spectrum for both chlorophyll A and chlorophyll B. But remember, this is a green pigment, right? Chlorophylls, both chlorophyll A and chlorophyll B are green, so they're not absorbing any green light. You see that? Little bit right at the bridge with yellow, but none of the green. So they're absorbing the blue, they're absorbing the red, and they're reflecting back the green. Okay? Do you see it? No? And then here's the action spectrum. So this isn't showing their absorption, it's showing the rate of photosynthesis. So how would you assess the rate of photosynthesis?
Increases in the blue and red.
To assess the rate of photosynthesis, you look at the rate of oxygen production, right? Oxygen is a product of photosynthesis, so you look at the rate of oxygen production. It produces oxygen most rapidly when it's given blue light and when it's given red light. Okay, which makes sense because those are the wavelengths of light they're absorbing best. Yeah, no.
Why is it with the far right side for the red that they don't absorb well?
Oh, on the far end of that red spectrum? It's a good question. It's the wavelength is just a little bit too large. The light's a little bit too low of energy. Too low of energy to get the electrons to either escape or too low of energy to get the electrons to, when they fall back down, to release light that can help a neighboring electron maybe escape. Yeah, it's a good question. All right. No questions? No additional questions? Thoughts? Okay. So the next question we need to deal with is this one. How do electrons flow through the photosystems? So the photosystems are complex molecular machines. Within those photosystems, you have pigments, chlorophyll, carotenoids, but the photosystems are very complex molecular machines because they have to do several things. They need to transfer electrons, facilitate the movement of electrons. They need to pump hydrogen ions against their concentration gradient. And so here's what's going on. You have chlorophyll A, one of our specific pigments, within these photosystems, pass excited electrons onto electron acceptors. So they split the electrons away from water. They split water molecules, releasing electrons. So when you get just the right wavelength of light, you can split that water molecule, take its electrons, and pass it to what we call the primary acceptors. The primary acceptors. Within these photosystems, we have this complex called the antennal complex. And within this antennal complex, it's just packed full of chlorophyll. Packed full of chlorophyll. And chlorophyll, again, it's absorbing sunlight. And then we've got a very special chlorophyll A in the middle of that antenna complex, or antennal complex, and it's called the reaction center. And that reaction center is the place in which the water molecule gets split, at least for photosystem 2, where you split the water molecule and you take the electron and pass it to the first electron acceptor. And again, what's the final electron acceptor going to be for the light-dependent reactions? NADP+, and P stands for plant. It still doesn't stand for plant, but it'll help you remember. And so we have two photosystems. We have photosystem 1 and photosystem 2. And I'm going to tell you something right now that initially is going to frustrate you, but when you remember it, you'll no longer be frustrated. Electrons go through photosystem 2 before they go through photosystem 1. And again, you're like, that's frustrating. They should go 1, then 2, because that's how we count, right? That's how we count. The issue is photosystem 1 can work independently. Photosystem 1 can work independently, and so you can have photosynthesis happening not using photosystem 2. And so that's one reason why we have to call photosystem 1 photosystem 1. It may not be first if you're using both, but in some ways it's more important because you can keep photosynthesis going without even using photosystem 2. Notice I said keep it going. You can't start to, anyways, we'll get there. The other reason is photosystem 1 was discovered first. Okay? And so it was discovered first, and photosystem 2 was discovered second. When they found a second, they're like, okay, we'll name the one we found first 1, name the one we found second 2, and then it just so happened that electrons go through 2 before they go to 1. It's unfortunate, but it happens. But now you'll remember. You'll remember 2 comes before 1, and it's unfortunate. Okay? So photosystem 1, the reaction center. And again, remember, this is a specialized chlorophyll A. This is the one that actually transfers electrons. This isn't one that just absorbs light and lets the electron drop back down and pass its light to a neighboring molecule. That's what the rest of the chlorophyll A molecules do. This is a special one. This takes the perfect wavelength of light and actually passes the electrons to something else. And notice photosystem 1, the reaction center, we put a number in there. We call it the P700, and P stands for plant. The P in here doesn't stand for plant either, but I remember that we need to have a P there because it's a plant. It actually stands for photosystem, but anyways. So P700, that's an interesting number. Do you remember that the visible portion of the spectrum goes from 400 to about 750 nanometers? So 700, we're like, is this a wavelength of light? And it is. This is the wavelength of light that the reaction center needs to pass the electrons out to the electron acceptor. P700. And what color is this light? Red. This is red. This is red light. And you're like, so what about all the blue light? Well, all the blue light then has to be absorbed by the neighboring chlorophyll A's and then passed around, right? Now, blue light's higher energy, yes? Lower wavelength, higher energy than red light. And every time an electron absorbs blue light, drops back down, passes its energy to a neighboring, some of that energy is lost, right? And if you have blue light and you lose energy and lose more and lose more and lose more and lose more as you keep passing that energy around, eventually what color is that light going to get to? Red, right? It starts blue, higher energy. You keep passing it around, losing a little bit along the way, and eventually it gets to red. And eventually it gets to red at the perfect wavelength to pass the electrons off. And so you see this light kind of bouncing around that antenna complex between chlorophyll A molecules until it gets to the perfect wavelength of light, hits that special chlorophyll A in the reaction center, and electrons get transferred. Yeah, Micah.
So you keep saying perfect, but this says optimally at 700? Doesn't that mean it's...
Well, that's if you get, so if you, what it means there is if you are only giving that plant 700 light that has a wavelength of 700 nanometers, it's going to, that's where it's going to produce the most ATP and the most NADH, because you don't have to pass around to get there. So that's where it's optimal, but that's the only place that the reaction center can work with is with this light. Now, if you give it any other kind of light besides green, because green it won't absorb at all, if you give it any other kind of visible light, it will absorb it and eventually it will get there. But it'll take some time, right? It'll take some time. Does that make sense? So that's optimally, that's if you only gave it that light, that's where its rate of photosynthesis is going to be greatest. But if you give it other forms of light, it still has to get here, but it'll have to pass around the chlorophyll A molecules to get there. Because initially it's too high in energy. But notice if we go back, I see your question, Levi, just one moment. If we go back to here, and Noah, was it you that asked why it's low here? Notice once we get past 700, it's just like, boom. Not very good at going through photosynthesis because the wavelength is too large to get an electron to transfer, and it's not high, the energy is not high enough to pass it around and to eventually get to the right energy, right? You can do that with light that's too high of energy, and then you're like, well, why is it lower here? And that's because it has to pass around before it gets to the right energy to split an electron. Right here is where it's going to be optimal. And then you're like, well, why is it optimal here when it's, because this is super high energy light and the plants are really good at absorbing that light. Really good at absorbing that light. So, but yeah, once you get past 700, it's just not great because the energy of that light is too low to pass it around and get to what you need. When you pass it around, it's going to get lower. And it's already too low to split the electron. Levi.
Yeah, I guess another question about if it can step that light down,
Yeah.
why cut out the middle of the spectrum if it can
You mean why not use the green light as well?
reflect that green light?
That's a good question. It's just it's the way it works. It's the way it's designed. I mean, if you had a pigment that could absorb green light, then it would be fine. Now, the thing you have to think about is this. If plants absorbed all of the portions of the visible spectrum, they'd be black.
Yeah.
And you're like, okay, well, I mean, maybe it would probably be that it would absorb too much light and now heat would be an issue. The tissues would get too hot too quickly, right? You put something that's black out in the sun and it heats up much faster than something that's green does. So there's a need to not absorb all of the visible spectrum. That would be my answer. And that's the way God designed it. Yeah. There's something calming about green, right? Green isn't green. I know red's not a good color. Red makes people angry. What colors is it that are calming? And is it blue? And then blue is in greens. So yellow and red make people hungry and angry, and that's why like a lot of foods places use reds and yellows. Because it makes you hungry, and then it also makes you a little agitated, so you don't want to sit there the whole time. It's subtle, yeah.
But is green calming because it's the color of nature, or did God make it the color of nature because it's calming?
That I don't know. Yes. Well, then you can ask the question of like, is it green because our brain tells us it's green, or does our brain tell us it's green because it's green? Like, is color actually is color independent of what our brain sees it as?
Is your green different
Right.
than my green?
That's the next. I don't know. All I know is my green. I don't know your green. Is your green different than my green? If you're red-green colorblind, then yes, your green is different than my green. Yeah. You don't even know what green is. You're like, I don't understand.
Actually, no.
Yeah, I know. You're like, I'm sorry. Yeah. Shh.
And there are plants that are like black.
Are there plants that are what?
That are black.
Sure, that are darker. Yeah, it's a good question. I don't know. I mean, there are plants that are much darker green than others, which if they're a lot darker green, they're reflecting different portions of the spectrum. They're probably absorbing more of that yellow light. And so you're getting like a really dark green and getting very little light reflection. But yes, it's a good question. Petals? I mean, petals tend to be... flower petals are different because they exist for a different purpose. Now, flower petals are modified leaves, but flower petals are existing primarily to attract animals. They're not for absorbing light for photosynthesis. Yeah. All right. Photosystem 2. Photosystem 2 has a reaction center, and look, its optimal light, the wavelength that the light has to get to in its reaction center is 680 nanometers. 680 nanometers, which is still red light. And it's slightly higher energy than this one. Right? Remember, there's an inverse relationship, so if wavelength is lower, energy is higher. It's slightly higher than this one, and that's because this one has a slightly more complicated task. This one has to strip the electrons away from water. This photosystem just needs to take the electrons from a low energy level and get it high enough to get it to pass to the next thing in the process. It doesn't have to strip them away from water molecules, but this does. It requires a little bit higher energy light. A little bit higher energy light. But what you'll see is that the electrons here, they get excited, stripped away from water, and then they drop a lot before they get to the next photosystem. And then they have to get excited again in that next photosystem to transfer over to NADP+. And you'll see this. I'll show you a map. You'll love it. You'll love it. I mean, you'll like it. So pigment molecules throughout the antenna corpuscles, we've already mentioned this, they absorb light and then they pass the light until it gets to the right wavelength in the reaction center. which is 700 nanometers in photosystem 1, 680 nanometers in photosystem 2. And so we call this process inductive resonance, where basically all these chlorophylls, if they absorb light that is not 700, okay, they'll pass it to a neighboring molecule, and eventually it'll pass around enough where it'll drop in energy enough to get to that right amount of energy, which happens at 700 nanometers for photosystem 1, 680 nanometers for photosystem 2. Yeah, no.
Can it pass green light?
They don't do a good job of absorbing green light. They absorb very little green light. They will absorb some, but they absorb very little green light.
Because I was just thinking you start at blue and you're going down, you just like get green light and passing.
Oh, sure. Yeah, unless it's like a big step drop, right? So you could drop all the way from blue to yellow. And that'll happen all the time. Yeah. It depends on where the electron started, where does it end up, and then how many levels did it drop all at once? Because if it moves from, say, the second energy level up to the fifth when it gets hit by a photon of light, and it drops from the fifth down to the fourth, then from the fourth to the third, then from the third back to the second, those are going to be much smaller wavelengths of light when it does a stepwise drop. Right? So if it absorbed blue light and dropped all the way from 5 down to 2, it's going to basically emit pretty close to what it absorbed, which is still going to be blue. But if it absorbed blue, went up 3 energy levels, dropped 1 energy level at a time, it's probably going to emit red each time it drops. Does that make sense? Yeah. So then you go from blue all the way to red with bypassing green. Yeah, because you can't really pass green light because they can't absorb it. Yeah.
So is that what's happening in both the photosystems, that it goes up and then it goes down?
It goes up and then it goes down? What do you mean? The electrons?
Yeah.
The electrons get excited, and then after they leave the photosystem, they go down.
Okay.
Yep. Yeah, you'll see this. You'll see this. So here's the photosystem. Here's this reaction center with our special chlorophyll, and here are all these chlorophyll molecules around. And you see here it's passing light through inductive resonance. It absorbed light that wasn't the right wavelength, and so it just passes it around, and when it gets to the right wavelength, it passes it here, and then that excites the electrons to get them to transfer to the next. You see? Boom, electron got excited, transferred to the next molecule. But it requires a very specific wavelength of light. Yeah?
So when it gets blue and then it gets
Yep.
out, where does it go?
Where does it? I'll show you in a minute.
Okay.
It leaves the photosystem and it goes to either plastocyanin or plastoquinone.
I was looking at it last night, I could really figure it out.
Yeah, I'll show you. I'll show you. Oh my gosh. Oh, wow. No, I don't want to do this. This one walks you through the whole process. I don't want to do this. I want to show you the process. Okay. So that one walked you step through step. What's going to happen here? I'm just going to show you a picture. And then if words work, like you like words better than pictures, you can go back and read that slide where it's step for step. Step by step. Okay, so here's where our electrons start. Water, right? That's our electron donor. So light energy came in here, and if it was the right energy right away, which is a wavelength of 680 nanometers, boom, it splits a water molecule, excites those electrons up to this primary acceptor. Okay? Then from there, the electrons can pass to the next electron acceptor, which in this case is plastoquinone. Now, plastoquinone, notice, is pumping hydrogen against its concentration gradient. You see that? Plastoquinone took that electron, used the energy that's released when the electron drops in energy to pump hydrogen ions against its concentration gradient. Just like what happens during cellular respiration, pumping hydrogen ions against their concentration gradient. When you see that, you need to think ATP. You're pumping hydrogen ion against their concentration gradient. We're making ATP. We're making ATP. Okay? So the electrons, so first thing happens, light hits here. When it gets to the right wavelength, or if it came in at the right wavelength, it'll strip water molecule, take those electrons, excite them to the right amount to get it here. Then those electrons will pass to here, plastoquinone. Then they'll pass on to here, another complex. Then they'll pass to plastocyanin, and then they'll pass back to another photosystem. Notice photosystem 2, photosystem 1. Photosystem 2 came first. Right? That's frustrating, because photosystem 1 should come first. But it was the first one discovered, and I already told you it can keep the process going. It can't start the process, because photosystem 2 is the only one that can split the electrons away from water. but the electrons can keep going, and I'll show you how that works in a minute. All right. So then they go to plastocyanin, and then they come back to this photosystem, and they wait until what happens? So now the electrons are here until you get that right amount of energy. You get that photon with just the right amount of energy, which is at a wavelength of 700 nanometers. And at that point, it can take those electrons, excite them up to this point, and then they can pass from here to ferredoxin, and then from here ultimately to NADP+. Yeah.
How does the electron get the right amount?
How does it get the right amount of energy? Because
Sunlight? Or?
yeah, if it gets anything other than the right amount of energy, it won't leave the nucleus.
Okay.
If it gets too much energy, it might leave. If it gives too much energy, it might leave the nucleus, but not go to the right place. Right? It just goes somewhere, and then ultimately would end up around some other atom. Okay. And then if it gets not enough energy, it'll get excited, but then it'll just drop back down to where it started. But if it gets the right amount of energy, it'll get excited, leave whatever atom it's associated with here, and go to some atom right here in the primary acceptor. And then from there, we'll transfer to ferredoxin, from ferredoxin to NADP+. Now, are you going to ask what do you need to know from this? Are you going to ask that? Oh, okay. Chris, and then Levi.
I was just gonna ask, so if I wanted to, like, shine a light on a plant and
Yep.
have it grow at its most optimum,
You'd give it red light.
give it, like,
Yep.
between
680 and 700 nanometers.
Okay.
And you can give it photons everywhere in between there.
Okay.
Yep. Yep. Now, somebody else could probably do just as well as you could, giving it only blue light, because blue light's not optimal, but it's such high energy that from a single photon you might get two that are the right, wavelength, So, yeah. I mean, so you got to choose either red or blue if you want, or you just give it white and then it gets both and it's just super happy. But if you're like, oh, I can only give it one color light, I don't know why you would be in that situation. But if you had to choose, I would choose blue, because blue light is calming and red light is not. Have any of you ever developed film and been in a red room? It's unnerving. There's something very uncomfortable about that. Levi. I think nobody develops film anymore, Dr. Engel. We have digital cameras.
No, what is, because it's like a green circle, but what actually is
The photosystem?
Yeah.
It's just a big, complex molecular machine.
it's just a solid molecule, basically? Or?
Yeah.
Okay.
Yep, a ball of chlorophyll A and chlorophyll B and some accessory pigments.
And so those are in the chloroplasts then? Or where?
Yes, these are in the thylakoid membranes.
Oh, okay.
You have that innermost membrane inside of chloroplasts, and they're in there by the tens of millions, if not hundreds of millions. I mean, they're packed in there tight. Yeah. Chris,
Sorry, so
why are you sorry?
I don't know.
You don't need to be sorry.
So the membrane of the thyroid complex, right?
Thylakoid membrane. What?
I'm just trying to figure out, like, the photosystems, they're held in the membrane.
Yes, in the membrane. Yes. Yep.
Gotcha. And so they're passing the
Here,
hydrogen.
let me show you this. There's the membrane with
Okay.
the systems.
Okay. and-
Still showing you the same process, although it gets significantly more complicated. You're like, there's so much more information there. And so that's why I hesitated to move here, but then I did.
Okay.
And so here's photosystem 2, here's photosystem 1, here's plastoquinone, here's plastocyanin, here's ferredoxin, all of the players we saw on the previous slide, you see? But now we're putting them into the membrane. And then you can see plastoquinone, when it's taking the electrons and transferring it, harvesting that energy to pump hydrogen ions against its concentration gradient, it's pumping them into the inside of the thylakoid membranes. And then now we've got, you see all these hydrogen ions? You see all those? We've created a very steep concentration gradient. Lots of hydrogen ions here, very little there. Where do the hydrogen ions want to go? They want to go out because of a concentration gradient and an electrical gradient. There's a lot of positive charge here and therefore a lot of negative there, and they want to go out, and they're going to go out fast. And when they do, you go out there, and this machine harnesses their kinetic energy or some of their kinetic energy and uses it to make ATP. Micah, and does somebody else have a hand up? Micah.
Can you explain again what plastoquinone and
They're electron acceptors. Now, plastoquinone, in addition to being an electron acceptor, is also a pump. It's also a hydrogen pump. Plastocyanin is not. Plastocyanin it's just an electron acceptor.
And then why are they named that way? Is there importance to that?
That is I'm sure there is. I do not know the answer to that question.
Just 'cause we were
My
talking
guess would
about
be,
blue and red light, and I see cyanin and think cyan is blue. Is that?
yeah, I don't know. Yeah, you'd have to go into the etiology. Etiology? Is that the study of names? Etymology? Etymology? You'd have to look and see who found it and then why they named it that. I don't know.
It just kind of collects electrons.
Yeah, I mean, it acts like some of those machines in the electron transport chain in cellular respiration, where it accepts electrons and passes them to the next machine. Now, some of those also acted as hydrogen ion pumps. The only one that acts as a hydrogen ion pump here is plastoquinone. Chris and then David.
Is that because most of the hydrogen is made from water? Is that...
Well, that's why it's located close to photosystem 2, because photosystem 2 is where you're generating hydrogen ions as you split water. But, I mean, although when you split water, you're getting hydrogen ions down here, and we're pumping more into here.
Do you get most of the hydrogen ions from the splitting of water, or is it just...
Yes, yeah, probably. Yeah, most of them are coming from the splitting of water, and that's why we only have one thing pumping additional hydrogen ions. Yeah. Yeah, David.
One more time, how is the hydrogen ions getting pumped against their...
Same way anything gets pumped against their concentration gradient. You have a molecular machine that harnesses energy and forces something against its concentration gradient. So in this case, sometimes, in a lot of our examples, we call that active transport. In a lot of our examples, the machine is hydrolyzing ATP and using that energy to pump it. In this case, it's harnessing kinetic energy from electrons, dropping in energy level.
Oh, so it's not using the ATP, but using the...
Correct, using the
okay,
electrons
okay.
falling. Same thing that we saw in cellular respiration.
Gotcha.
Yep, using the drop in kinetic energy in those electrons to fuel that pumping.
Thank you.
Yep. And ultimately, the electrons go to the final electron acceptor, which is NADP+, and that forms NADPH, okay, with 2 electrons and a hydrogen atom that it can take over to the light independent reactions. Okay. All right. I'm going to show you one more thing, and then we'll be done for today. I told you we might get through everything on Wednesday, and then we wouldn't have class on Friday. We did not. We're definitely going to have class on Friday. I don't know if we'll have to meet for the whole time on Friday, but we will certainly have class on Friday to finish this chapter. Sorry for giving you false hope, because I know what it means when you're like, we might not have class. You're like, sweet, after Gen Chem, I can go take a nap, right? Or you're like, could I take a quiz drop in Gen Chem and just never wake up, right, until like 3 o'clock in the afternoon? I get it, and I'm sorry for eliminating that question for you. So you're like, I don't care, Dr. Eng, I wasn't going to come to class anyways. Like, you can have class all you want, Dr. Eng, I'm not going to be there. All right. So here is photosystem 1 working without photosystem 2. And again, photosystem 1 cannot start the process. It cannot start the process. But once it already has electrons, so it needs the electrons that photosystem 2 split off of water. But once it has electrons, if the plant goes through a period of drought and it's not getting any more excess water, it'll keep this process going and it'll keep making ATP. Okay? It'll keep this process going and it'll keep making ATP. And so when the electrons get excited in photosystem 1 and they pass from this primary acceptor to ferredoxin, they don't go to NADP+. You see that? Which means the plants without water cannot do photosynthesis, but they can keep making ATP. So the electrons will go up there, we'll go to ferredoxin, then we'll go back to this cytochrome complex, okay, which will pump hydrogen ions against their concentration gradient, okay? And so you'll keep making, which will facilitate making ATP, but you can't go through photosynthesis. You can't actually make sugar, but the plant can keep making ATP. So without water, it can't make sugar, but it can keep making ATP and fuel what processes it needs to until it can get water again. Okay, and you're going to find this especially true in drought-tolerant plants. plants that live in really dry, arid environments that go prolonged periods of time without water. Well, one, they're usually pretty good at storing water, like succulents, right? If you ever had an aloe plant and you cut a leaf off and then you squeeze it and all this fluid comes out, you can drink it. Some people drink the aloe juice. It's gross, but apparently it's supposed to be good for you. So, but yeah, I mean, so a lot of them will store water, but they can keep going through this and doing this and keep making ATP in the absence of water until they get a chance where they can have water again, and then they'll complete the process and go through photosynthesis. All right? So that's another reason why photosystem 1 is called photosystem 1, even though it comes second in the process in what we call the linear flow, when the electrons flow all the way from water to NADP+. From water to NADP+. That's the linear flow. The cyclic flow, they flow from photosystem 1 back to photosystem 1, back to photosystem 1, back to photosystem 1. It's cyclical. It's not linear. It's cyclical. It's a circle, right? You just end up right back at the beginning every time you do the circle. Emily.
So do you know what air plants are?
Air plants?
Yeah.
I don't.
Okay, well they're plants that don't need like soil and stuff.
Yeah.
Do they just absorb water from like the air?
Yes. Yep. Yeah, so you're talking about like moss, liverworts, those certainly they don't even have roots, and there's no way to transport water. They pull water out of the air, and then they'll also, if you dump water on them, they'll absorb it that way. And so a lot of mosses and liverworts live in moist environments where water will condense on their surface, and then they'll absorb it that way.
Because you can't have a plant that just doesn't need water, right?
Correct. Yeah, because they can't go through photosynthesis without water. And if they can't go through photosynthesis, they can't make sugars. And if they can't make sugars, they can't nourish all the tissues in the plant. Because the only tissues that can keep doing this and keep making ATP are those that are actively photosynthetic. And not all tissues in the plant are. So this is, it's not like an endless process, right? You can't just keep doing this indefinitely because other parts of the plant are going to starve. And eventually the plant will die. Yeah.
So if it goes through the cyclic flow, does that mean that oxygen's not getting produced from the plant?
Yes, it does mean that. Yeah, because if you're not splitting water, you're not making oxygen.
Okay.
Yep, it absolutely does mean that. And you're like, well, then why does this plant even exist? If it's not making me oxygen, it's not making me sugar. Why do I care about this? This is like sad. This is a selfish plant, just trying to keep itself alive. You can look at it this way: it's trying to keep itself alive so it can make oxygen and sugar for you in the future. Chris.
So I'm just trying to understand, like, so we got the cyclical process, but
Yes.
we can't do it indefinitely.
Correct, and it can't do it unless it already had electrons to begin with, which came from water. So it can't just like if you didn't start the process, you can't keep this going. But so like if you split, let's say you split that last water molecule, and so it's going through the linear flow instead of finishing and going all the way to NADP plus, it'll just start going through the cyclic flow because you have no more water to split, and it'll just keep doing that to keep generating ATP.
Okay, but it doesn't. I mean, plants will pretty quickly without water, right? I mean,
Yeah, I mean, they'll die pretty quickly unless it's a drought-tolerant plant, but most drought-tolerant plants store water, so they still need water.
okay.
And it also depends on the size of the plant. Like if it's a small plant that most of its tissues can go through photosynthesis and keep doing this, it might survive for a while. Yeah. I mean, now do you want to do an experiment of how long you can keep your plant without water and it survive and still recover? Yeah. Do it. Yeah, Mia.
So like when plants are like losing leaves and stuff and they can't like absorb light anymore, like is it over for a plant if like all of its leaves are gone? You're just like, well, you know, rip.
Well, unless it's going dormant. Like so lots of plants will drop their leaves in the wintertime because the sunlight is not direct enough to make it energetically feasible to keep all those leaves. And then they'll just go dormant. Like they'll work on whatever energy reserves they have. Yeah, they'll usually come back, but some of them don't. Yeah, I'm sorry. Yeah. All right. Have a wonderful day. Enjoy the rest of your Wednesday. Those of you that have lab tomorrow, I'll see you tomorrow.